So let's get started. So now, today we're gonna talk about fluorescence imaging, especially fluorescence microscopy. This is one-day lecture, so you want to pay attention. I think today I'm gonna go through the basics of fluorescence imaging, and this is really relevant to those who are working for many biomedical and biological science, especially advanced imaging technique. You're going to use a lot of this. So even some of you are building and will make a new kind of fluorescence microscopy. And that is really, really making a big breakthrough in our biological research. And let's see why. So what we're going to talk about it is a kind of a groundbreaking work of this green fluorescent protein. And to understand what's going on and the principle of fluorescence, which is coming from the Jablonski energy diagram. And so that's coming from the understanding of the photons of excitation and emission. And the next, we will discuss a fundamental rule of this fluorescence, which is distinguishing how the exciting photon and emission photon, there's a differences in their energy or simply the color. And the differences in color is allowing us to block out the excitation so that you can only see the emission light or emission photons coming from the fluorophores. So this gives us an exquisite specificity, because you have seen a beautiful black background like a night light and like a sky in the stars blinking. So likewise, you will find out that fine information from this fluorescence light, and that's coming from Stokes shift. And the property of the fluorophores, the efficiency can be defined by the quantum yield. And we will discuss a general configuration of fluorescence microscopy and going into a little bit of more advanced topics. But the real advanced topic, the most up-to-date, will be covered at the later part of our class, our lectures. So let's first start with green fluorescence protein, or we call simply GFP, and there's so many now, so we call XFP, because there are so many kinds of colors available. So how many of you have used green fluorescence or fluorescent protein? Raise your hand. I mean, in some of your images. Okay, great. So not many of you. But this is such an important one, and that gave a Nobel Prize in 2004 for those three people, including Roger Chan from UC San Diego, who made a major discovery and engineering of this. So let's get started first. What is GFP, or green fluorescent protein? It's, of course, it's fluorescing, but it's a protein. And interestingly, we found this from nature, especially like here, a jellyfish. Whenever you go to the aquarium, take a look at jellyfish and that beautiful, beautiful colors coming from. That's where it gets started, okay? So because we identify and clone the gene of this GFP, and now we can utilize it. And let's take a look what the protein looks like. It's very interesting shape. Look at the size, it's a 3 by 4 nanometer. It's a tiny, tiny protein. But the real core is chromophore. Chrome means color. So the color generating entity is actually hidden at the middle. And it's surrounded by this beta barrel shape. So beta sheet forming a kind of interesting barrel which protects this fluorophore. That's why the green fluorescent protein is so powerful. Whenever you put into the cells and it expresses, it doesn't get to be affected too much by external. So it's actually quite stable. And as all proteins have a C and N terminus, you can find this, and there are this structure. So how does this work for change our biological, biomedical study? Because as we say, contrast is in everything in imaging, and the biological cells or tissues do not usually provide you contrast, especially biological tissue, except those of blood and or let's say the pigment, melanin pigment. Usually it doesn't have a color or provides a contrast. So here we go. We have this nice protein which you can engineer. How do you engineer? Hey, there's a target molecule of your interest. You're interested in this target protein. Then by the technology of recombinant DNA technology, you bring your GFP genes, connect them, that we call a fusion protein. Then you can actually recall that how this central dogma of molecular biology from this DNA level to RNA and protein, and this become a recombinant protein. But your focus is this target molecule. However, you realize target molecule doesn't provide contrast. So what you did is you actually tinker, like attach with your GFP molecule, and this GFP, you send out blue light such as 488 nanometer, and you will get emission light like over 5-10 nanometers. So now you have a tool to look at the GFP. Remember, you are interested in target molecule, but you can see GFP because it's connected. So this complementary DNA of target molecule fused to that of GFP. An obvious question is what? What would be your question from here when you are observing an imaging? You want to see the target molecule. Now you are looking at this fusion protein. Of course, you have to ask, hey, what happens to this target molecule if they are connected to this? Do they function properly? That will be your question. And luckily, so far, this GFP is such a small molecule and stable. It doesn't seem to interfere that much so far. However, I have to say some of other proteins, such as RFP or red fluorescent protein, it's known that that can be toxic to the cell. So you gotta be careful and always ask the question, whenever you have this tool, are we having no issue or no physiological changes due to this modification? That's what you want to ask question and prove it yourself or look at the literature. I have to say not all fluorescent proteins are known to be very much safe or not changing the cell physiology. So you gotta be careful for that. So of course, when you go to aquarium, you see this nice jellyfishes, and that's lovely. And now we have take the gene out, and then you put every cell of a fish. Now you can create this green fluorescent fish, which of course it's a good toy and you love it. And what's more, you know, as you can find in aquarium, there are many different kind of colors around for different kind of jellyfishes. So Roger Chen's group engineered this to make a palette of various fluorescent proteins. I don't even remember all the names now because there are so many, but let's go through just one thing which you want to know because you're likely to use in the future. For example, you will find GFP, but there's an eGFP. And this E stands for enhanced. Just remember that because now CFP or cyan fluorescent protein, this blue color, and EBFP, so all have an E because it's enhanced. And the other is M, which stands for mammalian. So M orange, so you can have orange color. I think the origin is from mammalian cells. And also there's a TD tomato. So it's a tomato color, but the TD is coming from tandem dimer. And also it's not here, but when we think about what about red fluorescent? Yes, the first red fluorescent protein is called DS red. And that DS coming from the name of this organism. And you gotta be thinking quantum mechanical way. Whenever you go to a little farther and longer wavelength, physically the fluorophore molecule has to be bigger from quantum mechanical point of view because it's a longer wavelength, the molecule's got to be bigger. So it's not single, but it's like tandem and dimer or tetramer, that means the molecule gets bigger. And from my own experience, sometimes DS-red labeling for a cell, the cell physiology might change. For example, the cancer cells labeled with DS-red, you gotta be careful looking at their own cell physiology is remaining the same or not. So that's interesting but important. So how come this fluorescent protein revolutionized our understanding of cell biology, let's say? Because now each of the protein, you can put different kind of protein. For example, you can label mitochondria with DS-red. This is RFP or red fluorescent protein. And tubulin, alpha and beta tubulin for mitochondria can be labeled in nucleus. Likewise, something like YFP or yellow fluorescent protein, and you can label it the nucleus, and even Golgi apparatus with cyan and mitochondria with DS-red. So this is such a beautiful one, and live cell, you can see what's going on in real time. And what's more, recall your immunology aspects. So you remember that we have a variety of infinite kind of antibody diversity that's coming from a random combination of different genes. Okay, VDJ recombination makes our antibody diversity. We took that idea from there. How about bring this similarly to some cells express gene of RGB protein? You mix them, but the proportion of these RGBs are different, then the result would be different colors. So with this, Jeff Lichman's lab from Harvard created this brain bow image. So because the neurons are next to each other, it's very hard to distinguish, but by using a random combination of RGB, you can actually create this kind of beautiful color distinguishing separate neurons. Okay, so all this power is coming from fluorescence. Let's go back to our history, how we find out fluorescence, that principle. There's a stone or like this. What's interesting is people figured out that if you shine this stone with, let's say, some kind of cellophane or colored ones as an excitation filter, which is shined only passes UV light. And on the other side, you will realize that, hey, there's some lights coming out. And if you put some, such as, let's say, blue filter to block this excitation UV, you can actually see there's a glowing ones of this fluorescent specimen. And of course, the rest will be no light, so the rest background will be black. So that's interesting. And how does this fluorescence coming out? So that emission, let's see, Suhyun, can you guess what the nature of this emission direction?
Isotropic.
Excellent. Yes, one of the points is once the excited molecule, when they emit light, it's isotropic. Every direction uniform. So that's very important property of fluorescence. And now we want to describe how this fluorescence happens as a principle from the quantum mechanical point of view or the molecular energy. So let's say this is a molecular atom and this is a electron. And when there's a external photons get in, if that energy of the photon matches to the next level, and this electron will go to the higher energy level, but soon this will come back to the original. And while doing this, that difference of energy will be coming out of this fluorophore. Okay, so that we can describe it as the ground to excited state, but this simple form of excitation state, in fact, has a multiple sub states. And whenever the energy goes up, and they actually tend to go down the lowest energy level of this excited state and come down as an emission. So the length of here stands for the energy. So you look at that, green is emission, blue is excitation. Which one is short? Green is short. because it lost some energy already. So short or smaller energy contains a photon which has a longer wavelength. That's the key aspect. And I want to see this a little more in detail. And that we call Jablonski diagram. So the ground state of S0 and excited state, there could be multiple kind of excited states. So when the excitation light coming in, it'll jump the energy state to the higher level, and they will undergo some kind of internal conversion, okay, or vibrational relaxation. And that sometimes can become a heat. The point is they lose energy until they got to this lowest energy level of this excitation state, S1. And then what happens? Because this is unstable, they will come down. Where do they come down? It depends, okay? It can come down to this a little different level or really down level of ground state. But that differences of the energy when they come down, they emit the light, and that emission light is we call emission. But sometimes it happens that that doesn't emit, but it just energy can be dissipated as a heat. Or if there's a lot more fluorophore around, between these energy transfer, they can just suppress the emission that we call quenching. Okay, so now because of this explanation, let me go through, let's say, Prahanu.
Yes, Professor.
So we call this fluorescence as the radiation of light, and that we call
The exact.
I can give you an example, either excitation or emission.
Oh, excitation.
Oh, sorry.
Emission.
Yes, emission by a substance. That's what we call
Fluorophore.
Exactly, because it fluoresces light, and that is as a result of incident radiation. Incident radiation is called
The incident radiation is excitation.
Exactly, and excitation was a shorter wavelength. Okay, so that Jablonski diagram is really illustrating the state of energy of a molecule, fluorophore, and transitions among them. So we call this as a excitation or absorption, because the molecule absorbs the energy and emission mechanism. And as I say, quenching is a process when the fluorescence decreases due to any process. For example, you will find, hey, I have a fluorophore molecule, like real dense, and you consider the more fluorophore concentration will give out higher fluorescence, but in experimental, you'll find sometimes it's not true, because when there's too high concentration, they actually self-quenching, okay? So it's not always holding that higher concentration of fluorophore. It's not always helpful for you to generate fluorescence. Okay, that's when the quenching becomes important. And now we will discuss about a person who observed this first time and put their name on it. Okay, so that's Stokes shift. So a simple words, I describe excitation wavelength and emission color, but in reality, it's a little more complicated. The molecule has its own energy state, that the excitation is not just one line in wavelength, it actually has a broad wavelength, okay? While the emission light also has a broad wavelength. However, the peak of these are different. In general, emission has a longer wavelength. Why? Because usually it has a lower energy. It doesn't usually have a higher energy than excitation. So higher energy, a lower energy of emission, having longer wavelength. So they're going to be shift. And this is giving us a tool to differentiate these two. But look at that, there's an overlap region. So this makes our life a little more complicated. So I bring this concept as excitation. You give an excitation, and then from excitation state, the state will go down to the lowest energy level, and then it starts emit. So this process of energy relaxation, you're losing some energy. That makes the color or energy different from emission and excitation. And that's discovered by Stokes, and so we put the name of Stokes shift of these differences of this maxima of absorption and excitation spectra. Okay, so the question here is obvious. Let me ask a question to, let's say, Sada. Let me ask you a question. Which one has low energy, emission or excitation in two-photon microscope?
Excited photon.
In two-photon microscope?
Exciting photon.
Yes, you're right. Two photon, that's true, but in general, not two photon in general, excitation, emission photon has a lower energy. Okay, so I think that I want to make sure you understand. Now I bring a little more in depth of principles of fluorescence. Okay. So this shows a little more complicated, but the reality, because when you are purchasing a fluorophore, you can Google and spectrum, excitation, emission spectra of a fluorophore. What you will find, not like simple clean form, but you will find this one. It's actually very confusing at the first time. So over wavelength, you see this dark black color is excitation or absorption spectrum. It looks like almost a mirror-like of a little shift is emission spectrum. This is the reality that makes our life difficult, but you got to have a question why this is the case. The reason is because lower energy has a multiple sub-energy level and excited states as well. So what happens is when there's light coming in to the fluorophore, all of these can actually excite the light. So for example, like this longer, high energy can excite, and that corresponds to this UV, and high. Just the y-axis is the probability gets highest or peaks at that level. Let's say in this case 488 because it's FITC ones. But at the same time, what about the emission? Emission actually happens. So you excite all this level, but the emission happens. They all come down to the lowest energy level of S1 or excited state. So you start from here. So in fact, there's no memory. What it means is when emission happens, the fluorophore forget about how it gets there, whether it gets from this or this or this current, they don't care because they start from reset of this 0 of S1, and then now emission happens. And where does the photon or energy state go? It can go anywhere of 0, 1 to 3. What that means is different energy level. So emission color is not single color, but different, but peaks at here. But because of these shorter ones and longer ones, you will see a spectrum. And what you notice, there's overlapping region here. That makes our life a little difficult because we call this as crosstalk. So you want to take a picture of green and red together. First, you take green and red, and you realize that these two images has actually overlapping regions. So it makes you complicated, and especially for quantitative analysis, it gets you into trouble, and the reviewers will ask you to prove that. So this is a real issue, and you want to always think about this when you choose a proper fluorophore, because in real biology, you don't necessarily see only one color. You want to see at least multiple color because you are interested in like interaction or structure versus a function with among different kinds of entities. So that's why this is so important to understand. And I want to think about the temporal dynamics. Okay, so first here, arrowing pointing up means excitation, okay? Down means emission. You already probably know. But I want to also explain you about this temporal aspect. So how long does it take for a molecule to go into excited state? In fact, it's super fast. It's like femtosecond order. So it's instantaneous. Why? There comes internal conversion, which means the energy gets leveraged lower and lower, or vibrational relaxation in terms of this molecule relax in terms of vibration. And that's a picosecond order. It's 1,000 times longer, but still very fast. But what are the determining factor of this here, fluorescence? You stay here for a very long time, I mean nanosecond order, and you drop down, giving us a fluorescence. So that is nanosecond. You want to remember fluorescence, this is a lifetime. All this process, what the dominant time bottleneck is fluorescence, which is nanosecond order. Of course, there are other things happen because there are inter-system crossing. It can go move on to another, and it can actually send out photon much slower, such as 1,000 times slower than fluorescence, that we call phosphorescence. In Korean, it's called ingwang, and that's what happens for dead bodies, bone, like in the dark, it can glow, and people are scared about it, like ghosts, and that's what this phosphorescence is about. Okay, so the characteristics, let's see, not. So let's go through one by one. Fluorescence light is red shifted. Okay, what that means? Wavelength. Not. Can you guess what this is?
Increase.
Great. Increase or spatial frequency.
Decrease.
Right. Relative to excitation light. That phenomenon we call stop shift. And primary origin is initial vibrational relaxation and subsequent fluorescent transitions to higher vibration energy level of the ground state. Okay, then emission wavelength are not only longer, but also, Suhyun? This is really important to understand. So emission wavelength is generally longer, but also with excitation wavelength, it is, can you guess?
Independent.
Yes. What that means is there's no memory. It doesn't care how we get into the excited, okay? Although the initial excited state is related to excitation wavelength, in the end, vibrational relaxation to the same intermediate state terminate the memory. I mean, there's no memory, means once it gets excited, it doesn't care how it get there. Okay, and the last one as a fluorescence light is usually, yes, this is actually pretty tough, so I will explain. If the excitation, even if you use a laser light, such as coherent light, in general, fluorescence is incoherent. What that means is each fluorophores, when they fluoresce, they do it kind of randomly, so you lose the phase relationship. So this is kind of important aspect when you do fluorescence microscope. So usually fluorescence from the samples are incoherent. So you don't necessarily use coherence, take a use of coherence easily. So that's kind of something you want to understand. All right, let's talk about key concepts in fluorescence. As light is observed and emitted with different energy when it undergoes some of energy loss. And this is what we call a Stokes shift. And a molecule undergoes an electronic transition, and that's explained in Jablonski energy diagram. Here your question is, hey, I have a different kind of fluorophore. I want to know about their efficiency of the fluorophore, whether GFE is better or like RFP is better, something else. For example, ICG, this is the only, not the only, only a few of clinically approved so that you can use for patients is ICG or indocyanine green. This is near infrared light. You are curious about the efficiency, and that you can think of, hey, there's a fluorophore, there's an excitation photon in. It doesn't always emit photons. So that efficiency you can define as how much of a fluorescence produce emission per absorption photons. So number of photons absorbed versus number of photon emission, that you can define as a quantum yield, okay? And quantum yield is pretty high for GFP, like 0.7. It's 70%, it's efficient. And even fluorescein dye, a typical and relatively economical and very useful one, has even 90%, but ICG has only 15%. So that definition of quantum yield. And the other thing, aspect, is a practical aspect. So we've discussed fluorescence process, and we realized different fluorophores have its distinct lifetime. And what the lifetime means, the average time of their remaining excited states before emitting a photon. So you remember previously I showed you out of this whole process, the timing, which one's the longest, except this phosphorescence, because that doesn't happen all the time. So out of this, you realize nanosecond. So this time usually dominates as a fluorescence lifetime. And this is a very specific for each different kind of fluorescence molecule. So that gives us another interesting contrast mechanism. It's an advanced technique called lifetime imaging. You can count lifetime and figure out where are the fluorophore. And finally, this is a molar extinction coefficient, such as when we learn from Beer-Lambert law, we remember that this extinction coefficient is how strongly a chemical species absorbs light at a given wavelength. Okay, that epsilon is defined as per unit length per unit concentration. So if you multiply by this concentration and the length, and you will get this absorbance. Okay. All right. And this is a reference. I just want to provide you information. Hey, when do we use this fluorescence? Are there any of endogenous fluorescence? The answer is yes. Our biological tissue and cells contain mitochondrial fluorophore, which is called NADH. It's actually abundant in cancer, so sometimes you can use it for cancer detection. And there's a NADPH. This is a reduced form. And interestingly, they have a different lifetime, 0.4 nanosecond, and compared to a longer lifetime when bound. So bound, unbound state has different lifetime. What it gives us an idea is by using lifetime, you can actually have a ratio of NADH and NADPH. That sometimes gives you another contrast. So when you are dealing with a fluorophore, what you want to really know is, hey, what's the excitation wavelength peak? And what are the emission? Usually emissions are longer than excitation, you can see. And these are endogenous fluorophore. Even collagen has its fluorescence at UV range. Elastin, FAD, NADH, and this is a amino acid tryptophan. You can see 280 nanometer, and still has a fluorescence. And even quantum yield has been studied. Okay, so far what I described at Jablonski, this is for your reference. I just want to reiterate you about just a new thing is a Raman spectroscopy. Here, Raman is you give a photon which goes to virtual state and then it emits, and that difference is over here contains the molecular information. So Raman is called as chemical imaging. On the other hand, the excitation takes only femtosecond, internal conversion and vibration relaxation picosecond, and fluorescence is nanosecond. So that's the lifetime. And so that's for your reference. Now I want to discuss about how to build a microscope with the fluorescence. In fact, once you know, it's very, very simple. It's as simple as this diagram. What it means, you have mercury arc lamp, so it shines light, and what you want to pick is kind of pick the right sample with the excitation. So you choose a blue light, and somehow this geometry is called the epifluorescent geometry. What epi means above. So you shine sample above, and you detect the sample image above. In that case, you can call it epifluorescent. If you shine from bottom and go through and detect the other side, it's called transillumination. So this is epifluorescence and very typical way. So how is it possible? You need a mirror that reflects this blue light, and sample will emit photons such as green and red kinds of photons. And somehow this has to go through to make an image. So this mirror is really strange. It has to be very special. Why? Lower wavelength, it works as a mirror, but longer wavelength, it works as it's just a glass, it just passed through. And then out of this, you can choose different emission filter to choose either green or red light. Okay, so that's what the epifluorescence microscope. How do you build? It's as simple because you just need excitation filter. This we call two different dichromatic or dichroic mirror. You need emission filter. That's it. Okay. How do you make it? You need a filter cube that contains these three different components of excitation, emission, or barrier filter and dichroic. And in fact, you can buy this unit, or if you are an advanced user, you should buy each of this by yourself. And then you have to know how to read this spectrum of transmission. So for example, excitation filter, red one, you choose this 480A, and then you have dichroic. This one serves as a dichroic, is a white color. So it reflects light the lower wavelengths, but it transmits longer. But you realize this one is actually not perfect, okay? Out of emission, excitation, and dichroic. So you need an additional one called the barrier filter, which provides this yellow color, and that makes an extra blockage of excitation light so that you can see emission. Now, guess what? You can see this is a cover of Cancer Biology book. You see a beautiful picture of a blue nucleus and red and the green of the cells, different kind of cells. So this is the power to provide which ones are cancer cells and which ones are not. So this is a fluorescence image, and fluorescence adding a specialty to the measurement. 정명? 정명, are you there? Okay, so can you guess what this? You cannot say. Okay, that's fine. And Laxman, can you guess what is special about this?
Specificity.
Excellent.
I know.
Right. That's it. That's the difference from the other kind of general scattering imaging. Fluorescence provides specifically which one you are looking at. So that's the key aspect of fluorescence. And these filters you will find out in your laboratory. You took out, there are various kinds of filter turret. You can buy this. This is actually very expensive, over $1,000, almost $1,000 for each of unit. You can have a multiple. You can have different ways of this filter block. But the point is, there are short pass filter which passes its lower wavelength. The long pass filter, usually fluorescence light is longer, so you use a long pass filter. But in the middle, you have to direct your excitation light, so it works like reflection. It's different from the other. Reflection and the longer wave, you pass through. That we call dichroic or dichromatic beam splitter. So that's what you need to have fluorescence microscope. I already show you here, but just remind you, you have epifluorescence, you have dichroic mirror shine on top of the sample and go through the emission and go to your imaging device. And now I want to, for just a couple of minutes, I want to go through something on reality. When you're working with fluorescence, what you have to be worried. You never want to put your sample on the light, room light, especially fluorescent sample for a long time. Always put aluminum foil or block the light, otherwise you'll keep losing it. So let me show you that phenomenon called photo bleaching. So when you're doing experiment, never put too high light when you're not collecting your sample, because you're already making change on your sample. The sample of fluorophore will bleach, which means it will not fluorescence anymore. Very interesting one, I brought it here. Look at, there's a 3-color fluorescence. You shine it multiple times, A, B, C, D, E, F. You see what's noticeable? The blue ones gets dim, gone. And then green ones are gone. And then later on, the red one remains, it will be gone as well. Why? The fluorophore molecule has a limited time of emitting photons, okay? So after, let's say, some green Phe molecule has only tens of thousands times of sending photon out, they will die, which means it will undergo irreversible photochemical destruction. It's not fluorescing anymore, okay? Because of this chemical damage or covalent modification, okay? So that's why, especially here, noticeable is shorter wavelengths or higher energy ones are gone first, okay? And how do you resolve this? You gotta save your exposure always. Save it for your actual data acquisition. When you are looking at the sample, don't use excessive amount of light. When you are not taking data, don't shine your sample. You are losing your sample. So that's something I want to mention. But on the other hand, people actually can utilize this property. And that's called, you can use photo bleaching as an active tool for studying a diffusion process in your sample. That we call FREP, or fluorescence recovery after photo bleaching. So let me tell you about this very interesting ways. So sometimes you are interested in, let's say, receptor distribution onto your cell. You are curious about how much of the receptor is diffusing on top of the cell. So for example, here, there are fluorophores on your cell membrane. And let's say you just shine a super high power laser onto here. So it blacks out, which means the fluorophores are gone. But you can think of, there are always diffusion on the cell, so the nearby fluorophores will diffuse in over time. So you can just check quantitatively what happens to this. In fact, this relaxation of recovery is an indication of how well they diffuse out onto here. Okay, so that's an interesting one. In this case, you can measure translational mobility of, let's say, this fluorophore, or this can label to receptors, and that receptors lateral diffusion coefficient you can measure. And I have to say one example that, like even tumor biology, you can do this, okay? And the other, I want to explain the last topic as a phototoxicity and damage. This, why also too excessive light is not good, because you can kill the cell. You know, vacuole formation, you can cause necrosis. So there are concepts of phototoxicity, especially, you know, fluorophore itself can be toxic too. So excited state fluorescence molecule can react to molecular oxygen to create free radicals. And that free radical is usually not good. It can damage the subcellular components, and it can affect physiology of the cell. So that you don't want to provide too much of fluorescence, because it can cause phototoxicity. On the other hand, photo damage is a little different, irrespective of fluorescence. It's like you go out to sun too much, your skin is getting old, and it's like that photo damage. Our mammalian cell can be damaged to UV light, high energy light exposure, because those high energy can damage your DNA and proteins, and that can damage your cellular physiology. So that we call photo damage. All right, so let me summarize rather fast. Today we went through all the fluorescence microscope, especially the tool for fluorescent protein, the principle of this understanding coming from Jablonski energy diagram, which leads to the differences between the energy of excitation from emission wavelength peaks. That we call Stokes shift. And you gotta know about the efficiency of fluorophore, that we call quantum yield, and how to design and build a fluorescence microscope, and something you want to be careful when you're running, keeping your experiment with the fluorescent samples. You've got to be careful for bleaching, phototoxicity. So we'll discuss about confocal next time. Okay, thank you for your attention, and I'll give you 3 minutes. You write down your questions, and okay, who are gonna do next time presentation? Is it Jeongmyo? Okay, next time Jeongmyo. All right, thank you for your attention.